So look at the amount of fibrosis. How do scientists measure fibrosis? These two pictures at the bottom, again, I think that's hard to see on the big screen. Maybe on either side it's more clear. The picture on the right shows a fibrotic heart, and you can see areas that stained kind of a bright blue scattered among the pink tissue of the heart. A normal heart is shown on the left, and you don't see those areas of blue. So there's a way of microscopically staining the tissue. You can tell how much of that heart has turned to fibrosis. Look at the graph on the left. The control heart has very little fibrosis, that small blue bar. The hearts that had chemically induced pulmonary hypertension had about 4 times as much fibrosis, the red bar. Animals that were treated exactly the same way except with the addition of Protandim, and you can see the red bar was dramatically reduced, significantly reduced to the green bar. So fibrosis was prevented. We, in this case, measured a specific gene product. This is a protein that signals your body to create scar tissue. You can see in the control, the healthy animals, a very small bar. The ones with pulmonary hypertension, a lot of signal telling the cells to become fibrotic, to create scar tissue. And you can see it largely blocked in the animals that were treated with Protandim. Protandim prevented dilatation of the heart. What does that mean? That means enlarged heart. It's not good when your heart enlarges. You might think a big heart could pump more blood. Not so. It can't develop the pressure because the contractility is not there around that small space. The hearts from these animals, the control hearts, you can see that blue bar represents the diameter of the right ventricle, the pumping part of the heart that sends the blood to the lungs. And the ones that had pulmonary hypertension, the diameter was twice as great. The heart was enlarged to twice its normal size. And you can see a significant reduction in the Protandim-treated animals. If you look at the number of apoptotic cells, dead cells in these hearts, the healthy control animals had measurable apoptotic cells. The ones with pulmonary hypertension, many more times the dead cells. And it looks like I forgot to put the green bar on that chart, but it's there. It's just reduced to 0. There weren't any dead cells in the animals that had Protandim supplemented diets. This is a measure of how well the heart pumps. It's cardiac output. It's blood, milliliters per minute of blood that heart pumps. And so if someone goes into heart failure, it means the pump isn't working. So a normal, healthy rat heart can pump 60 milliliters, a couple of ounces of blood every minute. The ones with pulmonary hypertension, that was reduced by about 40%. And in the Protandim-treated animals, you can see it's restored almost to the normal pulmonary cardiac output. This paper I didn't feature in an elite academy, but it's an important paper. It's a mouse model of another disease, and you might say, why Duchenne muscular dystrophy? There are only about 3,000 boys in the United States of America that have DMD, Duchenne muscular dystrophy. It's a genetic disease that only affects males. Muscles waste, they turn to scar tissue, they become fibrotic. So again, this is the recurrent theme you see in different models, different diseases, same mechanism in part. And what was seen here, that after 6 months on Protandim, these mice that have been engineered to have exactly the same gene defect that these boys with DMD have, they showed T-bars, lipid oxidation reduced by 48%. Does that sound familiar? The humans had a 40% reduction. Osteopontin was reduced by 57%. The previous VCU heart study also showed osteopontin reduced. That's the trigger, the message for scar tissue. And the MRI signal, magnetic resonance imaging, if you have a boy with DMD and you do an MRI, you can see changes in the muscle. and I'm going to be brief here. T-bars went down, osteopontin went up. Both of those were decreased in the Protandim-treated animals. And if you look at an MRI on a mouse, which is a difficult thing to do, these mice weigh about 20 grams, and the MRI machine was a tiny little MRI machine that probably cost twice as much as the big one that you go through.